analysis. So so as I said in the general session, we see a lot of opportunities when we have so many different modalities, especially the genetic data, and integrating with the imaging. There is a potential. The NIH actually has issued several programs on how to integrate genetic system biology into the brain study. But at the same time, there are a lot of challenges. So maybe, following the same theme of the sessions before, especially yesterday, we are talking about grand challenges, especially in the context of multimodal integration and analysis. Anybody want to start with? Maybe Tim, you want to start with this? How you view this?
Really, thank you for asking. I guess I see this, see a few different challenges. One is just simply getting good data from good research problems, right? Another is being able to sort of fluidly share that data in a way that this is a problem that we experienced when I was at the Mass General working in the Alzheimer's Center for many years, is just people are focused on their own data capture methodologies like imaging or genomics or whatever, and they have their own methods and so forth, and how to pull that data, bring it together, make it reusable, and really it's a data fairness problem inside the university. So I see that as important, and I guess another aspect of that, which you are very aware of, is explainability, analytic explainability, pre-modeling and post-modeling.
Okay. So my group has done extensive work on ADNI data. The ADNI data actually have imaging, couple of different imaging, and also have genetic side. They have when we try to merge this, two different data streams, try to come together to formulate a model for prediction of diseases. So there are quite a lot of issues. For number one, the most noticeable issue is missing data. So in different data stream, they have not every samples have all data. So how do you deal with such an issue? That gives us a lot of problem. So there are different imputation on this. And also when we have ADNI data, we also have this new NIH published, with UPenn on this ADSP. I'm not sure whether people know ADSP. It's a new NIA supported a lot for that data set. They also have all genetic data, mainly focused on genetic data and imaging data. But the two different cohort has different scales, different measurements. So there are a lot of trouble how to integrate them. At least those are the problems we had when we have to deal with these problems. I like to hear other, especially experts, how you think you deal with this. Yeah, Henry.
Well, I'm not an expert. I'm a learner about these things, but a critical reader of the literature, let's put it this way. And one of the things I can say that from our lab work point of view, we have a misalignment generally between genomics and proteomic data. So a lot of what we get out of patients is actually, it's not even genomics, it's basically a SNP analysis and, at least in our animal models, changes in gene expression rarely, predict changes in the proteome. And it's really not until we do both proteomics and genomics together that we can actually make inroads in terms of understanding disease processes. The other thing is that with the GWAS studies that are going on in various neuroscience areas, one can't help but always run into the same roadblocks, which is insufficient sample size. And in schizophrenia, for example, you're looking at 400,000 people being analyzed, and still the sample size being too small to show significant changes because of the fact that many of these diseases are polygenetic. I mean, they're obviously involving multiple genes with very small variations and whatnot. So what we definitely need, but I think, first of all, I think that bioinformatics and computational sciences is incredibly important to bring to bear in whatever we do here at UVA. But we have to be smart in deploying it on data sets that are unique to us, things that we can actually uniquely move something forward, whether that's in aging, whether that's in schizophrenia and autism, whatever it is. It has to be very focused rather than just relying on the incredible
I like to echo that. I'm so glad that you brought this topic up. Small data, I observe small data sets. It's a very common problem in medical domain. So each group is generating. Overall, you do have a lot of data, but I'm so glad you recognize among a lot of data, each cohort only have a small number of data. Okay, so in order to apply machine learning or deep learning approaches, this amount of the level of data is not sufficient to apply the common machine learning approach. So that comes to the new, actually there are new techniques in machine learning. It's called few-shot learning. And in few-shot learning, how to deal with just a small number, because in human learning, you don't, you don't learn based on huge amount of data, right? You just learn from a small sample. You're given even a small kid, you given an apple and a pear. Next time he or she can, point out which one is the apple, which one is the pear. You don't show a lot of samples. So that's how human learn. So actually machine learning has advanced this into how to learn from a small number of samples. It's called few-shot learning. So certainly I think, the transformation of the new machine learning models into the brain study is important.
I think Jake, Raish, and then Kevin were waiting to say something.
Well, thanks, Judy. My little hand was raised. I apologize. I need a bigger.
Okay.
Yeah. So, it's an honor to be amongst the expertise on this Zoom meeting. And I think that I'm dabbling outside of my comfort zone. So I work within the School of Education, and I oversee our concussion protocol at UVA for UVA athletics. And I've had conversations with Chris Holstage with Student Health on how we can apply that same model to our students who typically see around 500 concussions a year. With athletics, we see around 70 concussions a year. And we're one of those smaller data sets that we would love to leverage the expertise on this call. Now, granted, it's a very focused data set. However, we have a very captive audience. We see 250 to 300 athletes every year. And we had a similar conversation about, in terms of these data sets, what are the common data elements that we could collect across a particular idea, whether it's a mild traumatic brain injury or more severe brain injury, just for example. And then how do we leverage the expertise in this room in order to really push that forward and become prominent? Because I think working with Jack Van Horn on some initial work, as well as I'm looking across this room and what you're talking about with genetics, and that's something that unfortunately has been siloed from us, right? And I think this is a really important opportunity to say, we collect around 300 or so variables on every athlete that comes into UVA pre and post injury at several different time points. And this is just one example of a small data set that I would love to see this grand challenge open up the opportunity to leverage your expertise through machine learning, through quantifying large data sets. Even we have the opportunity for fluid biomarker collection at baseline and post-injury in order to get at genetics. And how do we incorporate neuroimaging, the clinical outputs, as well as more basic science outputs in order to allow UVA to rise to the preeminence that it deserves in this topic. So again, really an awesome group to be amongst, and I hope that we consider the opportunity to look at existing models to potentially yield meaningful contributions, both scientifically, but also clinically as well.
That's great comment. I think, I hope we can establish some collaboration to work together with such, such exciting research. So, okay, I have Kevin there.
Yep. Yeah. So part of my comment is just expression of a desire to collaborate in the sense that, so for example, we have about 500 people with autism imaged at multiple time points with both fMRI and EEG, and then we have whole genome sequencing data on them and gene expression data from peripheral blood. And then we have a group of controls as part of our Autism Center of Excellence. And so, one is just a desire to collaborate, and Tim and I have talked about this some as well. But then one point I want to make from that is the importance of the developmental trajectories as the phenotype that we're trying to understand. One thing that we've learned from doing some of the imaging genetics work with these groups and others, groups of patients and others, is that the longitudinal development sometimes is the thing that's most predictable or most informative in terms of trying to understand the interaction of the genetics, gene structure and gene expression in predicting the functional imaging data. And I think that can apply across disorders and into predicting individual learning profiles as well in typical development. So I wanted to mention that importance of a developmental perspective.
Yeah, that's great. So longitudinal time series analysis is very important in the brain-related research, right? So, yeah. So, Tim, you want to talk about something? Or?
So I just think all of this multimodality raises the issue of centerness, in that you want to be able to collect multimodal data sets in a coordinated way across a well-structured cohorts that are designed properly. I don't think it makes sense to try to collect data that's just sort of from random different people, right? So I think things like Kevin is doing, for example, are really, really important. And I think that is a key. You have to start with the notion of having structured cohorts.
That's interesting. So, David?
Yeah, hi. So I'm Dave Schlesinger, and I'm a physicist in the radiation oncology department, mainly at the Gamma Knife Center. So just, I guess my interest in this is maybe more from the clinical department perspective, but I think that mainly I just wanted to second what especially Jake and Tim have been talking about, which is like in the few of these Brain Institute meetings I've been to, it seems like a lot of the discussion really ends up focusing around what infrastructure do we have to somehow logically and in an organized way siphon all this data together so that we can then do machine learning and radiomics projects. And I just I just think that that is such an important focus, because I agree that especially within the clinical departments, there's not really any coordination of care that sort of lends itself to generating data that's organized in a way that we can then package it in the cohorts. And everybody tends to be siloed in terms of what questions they want to answer, which is sort of natural, but also in terms of where their interests lie. So trying to convince UVA to develop a system that we can feed all this data together, I think is really important. And as an example, I have a colleague named David Jaffrey, who's a Canadian medical physicist who's really well known in our space, but also a couple of years ago took a job as chief technology officer at MD Anderson. And I met him at a meeting a couple of years ago and got a cup of coffee. And he basically told me that when MD Anderson hired him away from Health Canada, basically his job was to come up with a way for MD Anderson to collect everything basically and feed everything through machine learning systems so that they're constantly learning from all of their patients all the time. And normally, of course, this would seem like a ridiculously sky-high dream of a job that he was given by MD Anderson. But if you look up David Jaffrey, you'll understand that like he actually operates kind of at the national level. Like in Canada, he was operating at the national level. So we have a lot of expertise at UVA. I'm partly on this call just because I wanted to meet all of you since you're all doing this data space, right? But there's just not a lot of coordination. And if we can solve that problem in this challenge, I think that would be a huge advance for UVA, because we all have pieces of the puzzle, but it's just disorganized. The other thing I want to add to that is specifically implementing machine learning and AI algorithms and data collection algorithms. There's also not a lot of coordination or sharing of the actual code base. So if there was some way of not having everybody reinvent the wheel all the time, I think we'd make more progress. And so for instance, for me, I mean, my job is mainly clinical, but I have a programming background, but not a lot of time to actually sit and write code. And so if we had some shared infrastructure as much as possible, and I know there's problems with that, and some way of both sort of advertising what's available and having some kind of version control system that everybody share basic infrastructure, I think that would help us also. Anyway, that's all.
I want to, thanks, David. I want to echo your point. Are you familiar with the recent NIH Bridge to AI program that call for data generation? So in the summer, Tim and I worked with other people intensively on one of these proposals. So I think your point has been recognized by NIH, because you do have to coordinate on data generation and get AI-ready data to be used for machine learning model. That's what they are calling for. They actually put $94 million onto this. So I don't know who will get funding, but a lot of proposals are submitted through the summer on this. Henry?
Yeah, to follow up on what David said, I mean, when we look in the mirror, we sometimes like what we see and sometimes we don't. And what I'm getting at is that the collection of neuroscience data from academic clinicians at UVA is relatively sparse. I mean, we actually lack, in most disease area, strong academic clinicians that have the luxury to actually be involved in collecting the kinds of data sets. So as what Kevin describes and Jake, they have great data sets. I mean, what Kevin has on his autistic kids is an incredibly valuable data set. We don't have anything like that even in epilepsy, where we're very strong. We don't have anything like that in Alzheimer's. We don't have anything like that in stroke. We have a sparsity. Here is a scan here and there and a few pieces of data, but we don't have the mindset right now in these departments that let's extract whatever we can get out of every stroke patient, out of every dementia patient, that we can actually analyze that in-house. So that's where I was getting at earlier. We got to deploy these tools on areas where we're clinically strong, hoping that we built up this clinical strength over time. But it's sometimes, I think Jaideep would probably agree. He's not on the call right now anymore, but that we have to be somewhat critical about where we have matching excellence in the clinical departments. There's academic excellence as opposed to, just delivery of care. And unless so for example, we talked about aging yesterday. We have an aspiring, a center for aging, but we are lacking the clinical excellence there and the ability to currently to amass all the data sets from these dementia patients longitudinally to feed into the kind of algorithms that we're talking about here.
Jaideep is here, so, and he wants.
Just get back, Jaideep, I stuck my neck out saying that, in many of our neurological disease areas, we actually lack the academic clinicians currently that will acquire the depth of data sets, that, for example, Kevin has on his kids with autism, that we wish to have across from stroke to, epilepsy and Alzheimer's disease, but we currently just don't have it.
Yeah. I mean, one of the big challenges for all medical centers, and you'll do the strategic planning, is to acquire clinician investigators. The pressure is to hire people who see lots of patients and generate a lot of money. And those are not the people who will generate the data you are looking for. So that's the challenge. But I hope your strategic plan would convince the leaders to invest in clinician investigators. That's what distinguishes institutions with depth, is having clinician investigators dedicated to really studying patients, not just for generating a bill, but for a study, which takes a lot more effort and time and organization.
Yeah, that's a
1 minute. You know what? I wish I knew all of you before the summer, because Jaideep, I wish this was all before the, so that Bridge to AI program was launched. I was looking for a group we can, write a proposal for data generation project. So we eventually went with the immunology people to generate immune cell data set. But Alzheimer, all this data can generate a good data generation project, actually, with all the clinical strength we have at the U.
I mean, and really, Aidong and Harry, it's a matter of joining the national AD system, ADRC system, and then we'll have access to a lot of their data as we contribute to it. And it's coming, but we have to enter those big systems. I mean, hiring Kevin gives us entree into the autism field, him and Micah. Hopefully we can join IDDRCs and have data coming out of there. And Alzheimer's, of course, we're knocking at the door to get in. But once we have entree, you would have access to larger and larger data sets. But we'll have to, price of entry is that we have to contribute our data to the national data sets and join in.